Hello, my name is Jocelyn and I'm one of the laboratory facilitators here in the Veterinary Technology Department at Douglas College. And today we will be discussing the different parts of a compound microscope. So when you are working in the veterinary clinic, you will be assisting your veterinarian by helping to identify microscopic objects, and we will be utilizing the compound microscope in order to do this. It's very important for you to understand the different parts of the microscope and their functions, not only to assist you with those organism identifications, but also to help if you run into any problems with your microscope that you can solve those problems in an efficient manner, which assists you with quality assurance. Now we will be going over the varying parts of the compound microscope. The first thing is the dust cover. So here at the college, all microscopes should have a dust cover placed on it after use. This helps to protect the microscope to ensure that no dust settles on it or any other things that might be kind of floating in the air. So we're just going to go ahead and remove the dust cover and place that aside. And then this is a binocular compound microscope, which means that it has two eyepieces here. And so all of the microscopes at the college, as well as out in the veterinary clinic, if you're working in general practice, most likely will be a binocular microscope, but you may see other versions as well. In order to turn the microscope on, we need to plug the microscope into a power source. So you will find the power cord at the back of the microscope, and you can go ahead and just plug it into the power source. Along the lateral aspect of the microscope is the rheostat. This is commonly called your on and off switch, but we will be referring to it as the rheostat here at the college. And so with this microscope specifically, you do want to turn the rheostat all the way on. And you will see that there were blue lights that kind of went all the way up of the microscope. We know that the rheostat is all the way on when all of those lights are illuminated. So now that we have our microscope on, we can go back to talking about the eyepieces. So the eyepieces here are actually adjustable, and so you can adjust them based on the shapes of your own eyes and whether or not you're sitting down or you're standing up to view your object. And inside each eyepiece is the ocular lens. So a compound microscope is made up of varying types of lenses to assist you in magnifying an image. We are going to move our way down the microscope. This area here is the tube. This just assists in connecting the eyepieces to the nose pieces of the objective lenses that we'll be talking about. So your tube is kind of just the connector portion of your microscope. And then heading down is the nose piece here. And so when you take your dust cover off and you're going to be using your microscope should always be set up to the 4 times, also known as the scanning lens objective, which is the smallest objective here. And so when you are going to change objectives, you will be utilizing the nose piece in order to switch to an increased magnification to the next objective lens, like so. And then going down to the varying types of objective lenses that you will be utilizing here at the college, as well as that you may see out in the veterinary industry. So the first one, as I previously mentioned, is the 4 times objective lens. This is also commonly called the scanning lens. And so this has the lowest magnification that we'll be using, and we always start at the 4 times objective lens when we're viewing a slide. The next objective lens is the 10 times magnification objective lens. This is also commonly called the low power objective lens. So you may hear that terminology used out as well. Next is the 40 times objective lens. This is also known as the high dry objective lens, but it is a 40 times objective magnification. And then lastly is the 100 times objective lens. This is also called the oil immersion lens. There is very important information to note about the 100 times objective lens specifically. Because it is commonly called the oil immersion lens, we do have to use immersion oil only when we are utilizing this lens. This helps to assist you in being able to view your image clearly. But please note that sometimes in the veterinary clinic, you will also see mineral oil around your laboratory area. these are not the same materials, and immersion oil is only to be used with the 100 times objective lens. The next part of the microscope is the mechanical stage. So your mechanical stage is right here. Your mechanical stage also contains your sample holder. So when you are placing a slide onto the microscope, you will be utilizing the lever within the sample holder here to gently place your slide onto your microscope. Next is the coarse focus or the fine focus knobs. I am going to be utilizing the coarse focus knob first, and it's very important to note that we actually only use the coarse focus knob only when we're setting up our slide in view at the 4 times objective lens. So I will be utilizing the coarse focus, and this moves the mechanical stage for us. So I'm going to be moving the mechanical stage, utilizing the coarse focus knob, and you can see that it moves that mechanical stage up and down in large increments. There is also a fine focus knob, which is located on the coarse focus, but it's on the more lateral aspect of the knob itself. And you may not be able to see it, but I'm moving it right now, and it moves the mechanical stage in smaller increments. So when I was talking about using the coarse focus only on the 4 times or the scanning lens, this is because it does move the mechanical stage in those large increments, and we don't want to break our slide or break our objective lenses. So when we are moving up objective lenses, after the 4 times objective lens, we will only be utilizing the fine focus knob. And so I'm just going to get my slide in focus utilizing the coarse focus knob, and I'm at the 4 times objective lens. So on the other side of your microscope, these are your stage controls, and there is a top control and a bottom control. If I move the top control, you will see that it moves the mechanical stage backwards and forwards. And then that bottom stage control knob, you will see that it moves that sample holder side to side. And so you will be utilizing these stage control knobs when you are viewing a slide in order to focus it and center it on your area of interest before you focus utilizing the objective lenses. Next is the condenser. So the condenser is kind of located within the mechanical stage and slightly under. You will also notice that the condenser has a kind of small circle of light. So what the condenser does is it takes the light from the illuminator, which is your light source at the bottom, and it condenses that light onto your area of interest within your slide. And there are other lenses located in the condenser to assist with that process as well. There is also a condenser knob that you can utilize to move the condenser upwards and downwards, and this again helps to focus the light. So your condenser knob is along near your coarse focus and your fine focus, and you can utilize this knob to move that condenser up and down to assist you with light control. Next is the iris diaphragm. So the iris diaphragm is located near the condenser underneath, and this is going to assist you with contrast. So the iris diaphragm has actually varying numbers written alongside that correlate with the objective lenses that you are supposed to use it with. So on the bottom part, if I'm going to go here, the first number is the 10 times that's located on the iris diaphragm. So what that means is I'm going to move my objective lens into the 10 times place, and then I can use that iris diaphragm to place it and match it with that 10 times, and that's going to assist you with the perfect contrast for your image. Next, if I were to move up to that 40 times objective lens, I would then use that iris diaphragm, move it up to the 40 times, and again, you may notice some variations in contrast. Please note sometimes you do need to move that iris diaphragm in between, but those values are there as kind of a base to assist you to get started, but you may have to play around with it. And if you are having difficulty with being able to visualize your image from a contrast perspective, please note the iris diaphragm, you may just have to adjust. After the iris diaphragm is the illuminator. So as previously mentioned, the illuminator is the light source. So that is your primary light source of the microscope that's going to shine up from the bottom through the condenser, through your objective lenses, through the ocular lenses into your eyes to assist you with viewing your image. So now that we have finished going through the parts of the microscope, I can utilize the rheostat to turn off the microscope. I would take my power cord out of the power source, place it in the back. There we go. Now that we've gone over the varying parts of the compound microscope, you should have a better understanding in regards to each part and its function. This will assist you in the veterinary clinic as well as here at the college when you are viewing microscopic organisms utilizing the compound microscope. It will also help you if you run into any problems with your compound microscope. You're aware of each part of the microscope and its functions to assist you with problem solving. If you do have any questions or concerns at all, please feel free to reach out to one of the laboratory facilitators during lab or after lab, and we'd be happy to help you.